I'm going to turn the lights out. If we were to stain that gel with amphibian bromide, we get something that looks like this. These are digested genomic DNA samples. And so this is why we have to do a blot. Just seeing the fact that, so if this DNA was not digested, what would it look like? What would this gel look like?
A smear.
Hmm?
I think you just have one long smear.
One smear.
Well, would you have a smear at all?
No, different.
It would be
Depends
on
on
the
the
top
top
together.
Right, so if it's not digested, those chromosomes are really big. So all you're going to see, I wish I was taller, all you're going to see is a massive, bright blotch right near the well, right at the top of the gel. Because if the chromosomes are intact, they won't even really run into the gel. So they'll just sit there in the well. So there wouldn't even be this smear. The smear is from all that digested genomic DNA. And you can see on this gel, there's no way to pick out a particular sequence. It's just this smear of bands. So this is why we actually have to do the blotting. So on Thursday, we're actually going to start doing a southern blot. You guys, we're not going to do the digest. The DNA is already going to be digested for us. So after electrophoresis, we treat our sample in situ. We actually soak the gel in hydrochloric acid, followed by sodium hydroxide. What are the functions of these steps? We're starting with hydrochloric acid. What does soaking the gel in hydrochloric acid do?
Depurination.
Right. So the HCl step is a depurination step. So you guys remember what the two purine bases are?
Adenine.
Adenine's one.
Guanine.
Yes, adenine and guanine. So the purines are the bigger ones, the double rings. And so depurination with HCl does not break the DNA backbone. And we don't completely depurinate our sample. So we are sparsely depurinating, which means we're removing just some of the A's and G's, just the nitrogen base part. This leaves the DNA backbone intact. So you end up so if we think of our DNA as this ladder, depurinating means the backbone's still intact, and we're going to remove some A's and G's. So we're going to remove half of some of these rungs. So it's not complete depurination. So why bother depurinating? Why is this important?
To loosen the DNA band so that it easy to denaturing.
Okay, that's one reason. Exactly. So, like what Swapni said, if you remove some of these hydrogen bonds, you've now made it easier to denature the DNA. That's one reason. What's another reason that we depurinate?
You get single strand.
Okay, so that goes along with it, makes it easier to denature. That's producing single-stranded DNA. Timmy, were you going to say something different?
No.
Same thing?
Yeah.
Nisha?
Stronger blotting.
Why?
I knew you were gonna say. The better bonding to the nitrocellulose. Stronger bonding to the nitrocellulose.
Right. So, and this goes along with what you're saying. So one of the other reasons is that, so it helps transfer, and I'll get to why in a second. So we depurinate with HCl. What's the sodium hydroxide for?
Denaturing.
Okay, so that's going to denature, but it also does something else. So
Hydrogen.
now we're going to separate this. Oops. We've already removed the bases from there. So when sodium hydroxide denatures, it also helps fragment the DNA. If there's an apurinic site, if there's a baseless site in the DNA, when you treat with sodium hydroxide, that will actually break the backbone there. So we'll actually get smaller chunks of DNA. And so these smaller chunks of DNA, they're going to transfer better, and because they're smaller, they're going to bind to the membrane better. So why is it important to denature the DNA in the first place?
To get the single strand, as it has to transfer and it gets complement as we add the probe.
So, last thing Scotty said. So the reason we want to denature our sample is because the way we're going to detect our sequence of interest is we're going to add a complementary probe to that. We're going to add a probe that's going to hybridize to a particular DNA sequence, which means we want our sample to be denatured so it can bind to this complementary probe. We'll talk a lot more about probing in a little bit. Directed. Okay, so after we've depurinated and denatured, we've kind of alluded to this, what is the next step? Exactly. So now, we don't actually work with a blot. This is the actual blotting part. We don't work with the DNA in the gel. We transfer it to some kind of membrane, basically like a sheet of paper. And so, you guys have worked with agarose gels before. They're messy, they're fragile. Transferring the DNA to this membrane makes it much, much easier to deal with and effectively concentrates it. Because if you look at a gel is a, I don't know what, centimeter high, half a centimeter tall. I'm very bad at judging distances visually. But your DNA sample is spread throughout the whole height of that gel. Whereas on a membrane, it's all concentrated on one spot right on top of the membrane. So it effectively concentrates your sample. So, couple key steps for Southerns are A, you have digesting your DNA, allows you to generate a banding pattern that you'll recognize later. And then the actual transfer, there we go, or blotting step, where you move your DNA from your gel onto a piece of membrane. And then we'll talk about the further steps in a little bit. How are your guys' gels doing? I'm going to turn the lights back on and shield your eyes if necessary. Don't touch them without gloves on, please. Are they set? Are they translucent? Where's yours? Oh. Okay. Yeah, I can